Okay, I think we'll go ahead and get started. So this is will be a quick tutorial of the tool that you'll be using this week. So this week we are looking at the gene expression. Where is your gene expressed and what tissue and under which conditions? Or so we're trying to see exactly when your gene is expressed, and hopefully that you can come up with a proposal about your gene and its function. So you'll go to the bar um, EFP website that will provide, and you just paste your gene right here, and you want to click these two boxes, and I'll tell you in a little bit why we do that. So you have here multiple data sources. So basically, those are different tissues of the plant, and they have measured the gene expression of all genes in those different tissues, right? And you have different databases. So you put your gene right here, click these two, then hit go. Okay, and the red color, right, it means it's highly expressed. The yellow color, it means it's low expressed. And you want to look at the key right here. So the maximum, it says 2000, right, 2100. So, and if that maximum, if that color is uh, between 0 and 50, then this is just noise, right? So they don't really mean anything, right? But because it's the maximum is here 2000, or if it could be 3000, it could actually go up to 48,000. This is the maximum gene expression on the chip that was used. So if this number is between 0 and 50, you cannot ignore these, right? Because it's just a noise. It's not really a signal. So you could see that it's highly expressed in the rosetta leaves. It kind of increases here, and then it kind of decreases in the senescing leaves, right, when leaves die. So, we picked this, right, to kind of mask all the genes that have, I mean, it kind of reads here, just to mask all the genes that show a standard deviation of more than 50% of the signal value, right? So, and this does the same thing, but for when we compare to, when we have a treatment and a control, as we'll see in a little bit. So you could learn some information from this figure, right? But then you could also check other other data. So when we say developmental map, it looks at the gene expression across different developmental stages. We could also, so there's also what's called development RMA. So this is also another, just an alternative statistical method for analyzing the data that comes out of the gene chip, right? I think it stands for robust multi-chip average. So it's just a different way to look at which one is better, which one is correct. I mean, who knows, right? So, but you could also pick abiotic stress. So abiotic stress is basically when the plant is under maybe any type of stress that is not biotic, right? Maybe drought stress, chemical stress, osmotic stress, and we want to look at it. And when you look, you see that all of them are red. It's highly expressed, which is, so the absolute value is maybe 1800. But this is not very helpful because I have two treatments. I have a control and I have a treatment, right? So this is a control and this is a treatment. And I want to have these be relevant to one another, right? I want that to be relative. So I would pick relative instead, right? And then go. So when you compare two things, it's better to pick relative and see what that. Okay, now I think it makes more sense. So this is the control. It is yellow, right? And yellow, it means it's not expressed, right? Or there is no fold change. This is the control. So the red color, it means there is a fold change, 2.8. So when it's red, it means 2.8 fold change. When it's blue, it means it has decreased. So this is the control, right? And this is the osmotic stress. You could see that it's here, it's blue, which means 0 after 24 hours of osmotic stress, right? Which is defined as adding 300 micromolars of mannitol. After 24 hours, I would see that there is, it says 1.4 fold change or 1.54. So there's a decrease in your gene under osmotic stress. And generally, the fold change, if you have maybe 2 or more fold change, right, that is important. If it's 2 or more, that's really high. If it's a 50 or 100 fold change, that is spectacular, right? So let's pick a different, let's look at embryo, see how my gene is doing, how is that expressed, and let's pick absolute because it's not really comparing, there's no control. So let's just pick absolute. And you could see that kind of you're developing embryo, it kind of increases during the development of the embryo, but then it decreases, right? But again, you see that the value, so this is the absolute. So the absolute value, right, it should be more than 50. If it's less than 50, that is noise, right? We could see that's 200, almost 300. So that is a real signal. So your gene might be doing something with the seed development or the embryo. We could actually look at the seeds. There's also another database for the seeds. So let's see what your gene is doing. So you want to pick absolute, just to make sure. Okay, you could see, you'll actually have to read each. If you see that one figure of those is interesting, then you would read and see what that means. So seed, let's look at germination. When your seed germinates, if your gene is doing something. Oh, okay. So you could see that it says, so the absolute value is 400, which is really highly expressed. Yellow is 0. So you could see as the seed starts to germinate. So when you put the seed in water, so stratification is when the seed starts to get water, right? And you could see that your gene is really increasing as the seed germinates. So you could say that your gene has something to do with germination. So you have 20 databases right here, right? So what I don't want to see is that you go and take a picture of each one of those databases, right? That is not necessary. You only need to take maybe 2 or 3 pictures, 4 pictures of the databases that are really cool, right? And that give you useful information. So yeah, so you don't really have, but go check them quickly. You don't have to obsess with any single one of those unless you see like really cool data. So, and hopefully that you would come up with an idea of what your gene is doing. So this is one website, but another website is, so the one that we just explored shows you where in the plant your gene is expressed and under which conditions, drought, cold stress. But this website tells you where in the cell your gene is localized. Right? So you put it here, look up. Okay, you see that it seems to be highly expressed in the chloroplast right here. So let's click here. And you can see that it's really highly expressed, that red color. This is like the maximum expression. So So this is something else that you could do. So yeah, by the end of the day, you should be able to come up with a hypothesis, or you should propose a function about your gene and what it's actually doing. Okay, hope this is helpful. See you next time.